Good afternoon, everyone. Thank you so much for joining us today. You are joining the Native Fuse Alliance Student Showcase, which is a virtual platform in which we want to share all the amazing work that the students are doing in food, energy, water systems. And this is a new series that we would like to develop into the next several years. And before we begin, I want to introduce myself. My name is Carlotta Chief, and I am a professor and extension specialist at the University of Arizona. I'm the lead PI for the University of Arizona within the Native Fuse Alliance, and I'm also the director of the Indigenous Resilience Center. The vision of the Native Fuse Alliance is to support the pathways of Native American students in food, energy, and water systems, as well as to address the fuse challenges that Indigenous communities face, and to support students to find ways to work with their communities to co-design solutions to these challenges. The Native Fuse Alliance is a partnership with the American Indian Higher Education Consortium, University of California, Berkeley, University of Arizona, and many tribal colleges and Native American organizations across the Western United States. This program is funded by the National Science Foundation. We are in year 2 of 5. And we are building upon a previous training program that both UC Berkeley and U of A had, which is a National Science Foundation National Research Traineeship. And it's at the University of Arizona, it's called the Indigenous Food, Energy, Water Security, and Sovereignty. And so we're really delighted to be here with everyone today on this virtual platform, as well as for our students to share the work that they have been working on. And we hope that this will be one way that we can connect our students across the Native Fuse Alliance. And so we're really excited to kick off this season and to have you here today. And we will be having our annual gathering of the Native Fuse Alliance, March 22 to 24th, in which our students can continue to network and have discussions around their projects, as well as connect with partners across Native Fuse Alliance. So welcome again. Thank you so much for being here. We have 2 IndigiFuse students who will be sharing their work. The first one is Luke Presson, and the second one is Chrisa Whitmore, both doing work along the food, energy, water nexus, and both who have really contributed to educational partnerships and development with Diné College, as well as Star School and many of our other community partners like Six World Solutions. So we're really excited to hear about their work and to share it with all of you here. I'll go ahead and give it back over to Torin. Thank you.
Okay, thank you, Dr. Chief. So we're thrilled to have both of these students here, and I will turn it over to Luke Presson.
Okay, thank you, Torin. I'll go ahead and share my screen. Okay. So, hi everyone. My name is Luke Presson. I'm a PhD student here at the University of Arizona. I'm in the environmental engineering department, and I was an IndigiFuse trainee starting, I believe, in the fall of 2021. So it's been a fun journey. Today, I'm talking a little bit about my research in membrane distillation for potable water reuse. The lab that I work in does a lot of water treatment technologies, mostly membrane-based water treatment technologies, and we really focus on water reuse. So turning wastewater into drinking water, basically. So I want to start with just why potable water reuse. We do a lot of non-potable water reuse already, especially in Tucson. And what I mean by that is So a city, Tucson, for example, generates quite a lot of wastewater, and that goes to the wastewater treatment facility. And most modern facilities can treat that water to a pretty high quality level. We call it typically like tertiary treatment. And that water is often used, or at least able to be used, for things like urban irrigation or agriculture or even cooling. And that's what you'll often see in like purple pipes, for example. So a lot of the times you'll see purple pipes in someone's yard or something, and you'll see a sign that says this property uses reclaimed water or something like that. So that's non-potable water reuse, which we already do. But I want to talk a little bit about potable water reuse, which is basically treating water to that level of quality that you can be used for irrigation. And then beyond that, that you can actually use it for drinking water or in your house for showers or anything like that. There's some, of course, obvious reasons why you might want to do potable water reuse. Number one is it provides more water to be used for any purpose, right? You can use potable water for urban irrigation and agriculture as well, but you can also use it for like I said, showers or drinking or anything like that. And you also don't need a completely separate piping system. And since there's not quite as much demand for non-potable water in cities, for example, a lot of that water ends up just being discharged into the environment. Whereas for potable water reuse, we would really be using a much higher percentage of our wastewater. And so I don't want to talk too much about the need for more water availability, because I think that's probably pretty high on everyone's mind. But just that potable water reuse is really important. And it's also much cheaper and much less energy intensive than alternatives like seawater desalination or even importing water. And then I also want to talk a little bit about membranes. So like I said, my lab focuses a lot on membrane-based treatment. And so these are a couple examples of some treatment trains. And let's see, we're not moving forward. There we go. And these membranes that I've highlighted here, especially reverse osmosis, are very commonly seen in potable reuse treatment trains. They're not necessarily critical, but they're usually like the keystone part of any potable reuse treatment. They're very good at a lot of things. One of the key reasons that most places absolutely will need something like reverse osmosis is for salt removal. So for example, the water that we get in Tucson, at least, is from the Colorado River, which has pretty high salinity. So something like 700 milligrams per liter of total dissolved solids in that water. And then after municipal use, this is kind of a rough estimate of a number, but you'll often get something like an addition of 400 milligrams per liter of TDS or salt. And if we want to reuse that water, we really have to get some of that salt out, if not all of that salt out, or it'll accumulate too much to be able to not be used anymore. That's where these membranes really come into play, and are basically required for potable reuse in places with fairly salty water like ours. So most membrane treatments are pressure-based, especially the ones that most people are familiar with. So things like microfiltration, ultrafiltration, and reverse osmosis are the big ones. Basically, think of it as membranes with fairly small pores that you're just like pushing water through, and the water goes through the pores while most of the contaminants that you don't want are stuck behind. So microfiltration and ultrafiltration have larger pores, so they can reject some of the larger things like particles and viruses. Whereas once you get down to reverse osmosis, you're actually rejecting like salt species like sodium and chlorine. Those are also very widely used today. So they're actually commercially viable today, and are being used quite often in like seawater desalination or other water treatment plants in the United States and actually around the globe. The one that I want to talk about today is membrane distillation, which is a fairly, or at least relatively new technology, and it's still mostly in the pilot phase. And it actually uses a completely different mechanism to purify water. So it's a thermal water purification technique that I'll talk about here with this fairly simplistic diagram here. So if this is the membrane in membrane distillation, you've got these pores, and on either side of that, you might be running one water that's your feed side, and then another water that's your condensing stream, that's your purified water. And the membrane has fairly large pores, but the key component here is that the membrane is hydrophobic, and so water really does not want to contact the membrane, and the water will not flow through these pores in liquid form. So without any temperature difference, you just have two waters flowing without touching. If you heat your feed stream up, even if we usually operate at maybe like 70 degrees Celsius, so it's below boiling, that water will actually start to evaporate, and instead of traveling through the pores in liquid form, it'll travel through the pores in vapor form. And since things like salt or other compounds in the water cannot vaporize at those temperatures, you actually get really clean and pure water on the other side of your membrane. So membrane distillation has really capability to purify extremely contaminated waters to really high quality. And it also, one drawback is that it's a little bit energy intensive. Heating up water is of course much more energy intensive than just using pressure. So the applications for right now are a little bit niche. Ones that I will show. The one that we use is in conjunction with reverse osmosis. So one thing that I didn't mention about RO is that it's great at treating water and produces very clean water on the other side of it. However, it also produces this concentrate stream. So if you use reverse osmosis to treat some wastewater effluent, I'll put some numbers here. It might treat 60 to 80% of that water, and you get all of that as pure water. But then about 20 to 40% of that gets rejected, and is now that much more concentrated. And that water is very difficult to deal with. It's so concentrated that you can't just dispose of it into a river, for example. So something needs to be done with that water, and that's where membrane distillation comes into play. We try to use membrane distillation to treat that water and create more drinking water while also minimizing the volume that needs to be discharged. And part of the reason we use MD is because it can tolerate much higher concentrations than reverse osmosis. So when we put some numbers down here as well, the concentration of this reverse osmosis concentrate is quite high. It's up into the thousands of milligrams per liter of total dissolved solids, also with quite a lot of organic compounds in it. So membrane distillation is able to treat that pretty well. So I want to talk a little bit. I'll go through this quickly. Engineers love these schematics, but I'll go through this very quickly. So this is a bench scale design, which is a very small system that's used to research kind of like theoretical aspects of the process. So right here in the middle of that white line, or that white space, is the membrane. And then on the right side, you have the feed tank that's basically just circulated. And then on the other side, you have the distillate tank, which is also circulated. The feed is just heated up and is our dirty water, in this case, reverse osmosis concentrate, and is purified by that membrane in the middle. And this is what that looks like. This is a picture of my bench scale system, just to show you guys scale a little bit. This piece right here, I'm not sure if you can see my pointer necessarily, but that piece on the top is the membrane. So it's quite small. And just recently, we were able to actually publish this paper. So this is my first author paper that I was pretty excited about, and really focuses on fouling of membrane distillation with this water. Fouling being when contaminants in the water basically stick to the membrane and start blocking those pores, which is really detrimental because you start producing less water and your process becomes really inefficient. I won't go through the technical details because we're fairly time limited here. But the concept is basically that we found the organic compounds in the water don't really cause much, don't really cause any problems. Whereas the
perfecto.
inorganics,
Sí, estoy aquí con los, puede llamarme en cualquier momento cuando tiene que estar despierto.
whereas
Sí.
the
Bueno, en
the
este momento no,
inorganics-
ellos están en la escuela, pero Julie está con Daniel.
Oh, Connor, if you could mute your mic.
Brownie, yo estoy aquí con los niños, mis bebés.
Whoever
Okay.
is the meeting host can mute for him.
Sí.
Torin, I think
So-
you can do that.
Yeah, it's muted. Sorry about that.
No worries. So yeah, we really just found that the inorganics are the main problem with this water. And so when you're trying to maintain a high water production rate, you really want to focus on things like calcium, and you don't have to focus too much on the organics. So that's what we've done in the past. I'll move on. And then we also found that you can clean the membrane pretty successfully to recover some water flux. Water flux is the term that we use for basically how quickly you're producing water, or your water production rate. And so we found that before you do any cleaning, if you run it for a while, your flux really goes down, but you can clean it with things like acid and recover a lot of that water flux. So the next stuff that I want to talk about, which is really what I'm starting now, is a pilot scale membrane distillation system. This is connected with a MBR, which stands for membrane bioreactor, which basically is a miniature water treatment plant. So we take wastewater actually from the Tres Rios water reclamation facility. We take raw wastewater, treat it with the membrane bioreactor, and then treat it again with the membrane distillation. So this right here is the membrane distillation system. That's really what I'm focusing on. But what you get in here is, with this system, you send in raw wastewater and you get out really clean, potable water. So I took some pictures of our system. This is what it looks like. It's inside the shipping container, which if you peek in here, you can see the membrane distillation system. And then from another angle, you can see most of it, where these three cylinders are the membrane bioreactor. And then that blue piece in the back is actually the membrane distillation setup. So I show this again for scale. You can see how much bigger this system is than the bench scale one that I was running earlier. It's designed to treat about 1,000 liters per day is really what we aim for. Again, another schematic. I won't talk too much about this. The only real difference that I would focus on is there's a channel in here. AGMD stands for air gap membrane distillation, and you actually have a channel in here that is the distillate or the permeate that's completely separate and actually is produced inside basically an air gap with a vacuum applied. That's all I'll really say about this, but I'm happy to answer any questions about it when we have time. So the objectives of this part is to maximize production. Like I said, we're aiming for about 1,000 liters per day, and we just want to see what parameters we can tune to get that level, and then maintaining high water flux with like cleaning the membrane and things like that. We're still kind of at the beginning of this phase, so we've really focused on this maximized production so far, and really also just the flow rate is what we've found. So if you look on the x-axis here is flow rate in liters per hour that we're just circulating the water through the membrane, and then you can see over at higher flow rates we get higher water flux, so faster water production. And then on the other axis is specific thermal energy consumption, which is really just a measurement of energy efficiency. So how much energy is needed to produce a certain amount of water. So that also increases with flow rate. So it's a bit of a trade-off. We have to decide which of these to use, whether we want to operate at high water production, even if it's taking a lot more energy. And then the other thing is this water or this membrane process has a rejection. This is the units here might be a little bit unfamiliar because it says log rejection. Really what that means is we're getting at about 99.9% rejection. So if our feed has a conductivity of something like 2000, we are actually getting our permeate conductivity is below 1. And so just to put that in perspective, our tap water is at about 700 conductivity. So we have really, really pure water coming out of that system. So there's a little bit more research to be done. I'll stop there just for time, and wanted to thank my advisors, Dr. Hickenbottom and Dr. Akili, and then some of the students or even graduated students that I've worked with for this project. Dr. Mukta Hardikar, Vrinya Felix, Jeb Shingler, and Marco Malaguzzi, as well as Dr. Armando Durazo, who helped a lot with the instrumentation, and then just the rest of the Her Art Lab that's been really supportive, as well as some funding sources, of course, one of which is Indigifuse, which I'm very thankful for. So I'll open up for questions there. Hopefully I'm doing okay on time.
Yeah, great. We have about 4 minutes. Thank you so much, Luke, for kicking this off. It's so great to hear from students. So if you have a question, you can unmute yourself and ask it, or just write it in the chat. And I don't know if you saw the shout out from Ariana where she said, I need to head out to another meeting, but great job, Luke. And I can't wait to see the rest of the presentations throughout the next couple of weeks. Thanks. Anybody have a question you want to put in the chat or ask?
Luke, I have a question. What, when this is ready to be in a community, where do you see this? East Coast, Southwest, other countries, like who's going to be using this?
Yeah, so it's kind of a specific component or a specific technology that's really useful for extremely concentrated waters. So things like a reverse osmosis concentrate, that's probably going to be used mostly in the West, just because of the water issues here, and also the high waters here are typically a little bit more salty. It's also really useful for brine from seawater desalination as well. And then finally, the real use for it is, since it's a thermal process, you can often use like waste heat from an industry or something like that. So you can heat up the water. You know, there's tons of industries that create a lot of waste heat, so you can often use that. And you might pair this with some kind of industry like that.
Awesome. Thanks, Luke. And there's a question from Dr. Chief. She has her hand raised.
Yeah, thank you so much, Luke, for sharing your work. So yesterday we were at Diné College meeting with Dr. Shazia Hakim about the same topic about water reuse and also how that fits with agricultural production. So my question was really how Indigifuse has influenced your work in terms of your perspective on this type of water treatment approaches for rural or indigenous communities. Could you share a little bit about that influence in your training?
Sure, yeah. Yeah, I mean, I started this out a little bit not knowing how membrane distillation, for example, might be useful in I'll say more rural or off-grid applications. I think through Indigifuse, I've interacted with a lot more students and thought a little bit more about kind of fit for purpose water treatment. And so I still think that membrane distillation has some limited applications for off-grid treatments. You can use like solar energy to use it, but typically there's better options. But the traineeship has really taught me a lot more about, yeah, I guess I'll come back to that fit for purpose treatment. So again, my lab does a lot of membrane work. And so while I focus a lot on membrane distillation, we also use things like nanofiltration, which is much better applied to communities like the one that we actually went up and visited with the solar nanofiltration unit. So I think the big takeaway is just making sure everything is fit for use, and you're listening to what the community needs rather than just trying to fit like my own research or my own application to anything.
This is great. And I want to make sure to give Chrisa enough time. There's just a great question that was put in the chat by Ronaldo Sosi. I don't know if you want to address that quickly, or maybe if there's time at the end, we can circle back.
Yeah, I'll do it really quickly. So the question's about using solar energy. We definitely do. There's another student in my lab that has a system that uses concentrated solar power. It's not my work, so I won't try to speak to it too much, but it's definitely an option. Yeah.
Okay, great. Thank you so much. Really appreciate it. And so we will turn it over to Chrisa Whitmore.
All right, I'm just gonna share my screen. Okay. Hi, everybody. My name is Chrisa Whitmore. I am a PhD student in the Biosystems Engineering program, and my advisor is Dr. Barry Pryor. And I am looking at mushroom production and light and substrate composition to see if we can improve the nutraceutical content of our product, which is edible mushrooms that we are growing. So just for me, for organization, pretty much this is what this slide is giving me. So I'm just going to talk about background information because I know everyone's not familiar with growing mushrooms. So I'm going to give you a little bit of background information, the objective of my project, as well as like my methodology. And we're going to go into a little bit of my data as well and next steps, of course. So bioefficiency so these are some of the terms I'm going to be talking about throughout my presentation. So bioefficiency is pretty much just a calculation that farmers use to determine what is the most effective substrate for a specific mushroom species. So it's pretty much just a calculation of the weight of your entire harvest divided by the dry weight and multiply it by 100. So it's a percentage. And nutraceuticals are something that can help with providing like protection against like chronic diseases. And I get a lot of questions that kind of it's like, okay, what is the difference between nutraceuticals and supplements, right? So supplements are just single-use substances that you need to add some other like micronutrients that your body needs, whereas nutraceuticals come from a food or a part of a food, and it can rarely cause like side effects, and it's pretty much just nutrition and medicinal benefits. And flush is pretty much just a harvest of mushrooms. So when you're cultivating mushrooms, you have one big first flush, which is first harvest, and you sometimes get a second flush as well. So those are the terms I'll be using. So everybody asks me, why mushrooms? Why did you decide to go into growing mushrooms? And it's because they are a wonderful benefit to everybody's diet. They are about 40% protein by dry weight. There's a lot of antioxidants within mushrooms, and they provide a really wonderful source of protein and antioxidants, and you get a wonderful, awesome benefits. They are also natural decomposers. So you can pretty much just use any sort of agricultural surplus byproducts. So you can use like straw or sawdust or cottonseed hulls, and you can grow mushrooms on that as long as they're sterilized, of course. And so there's a lot of benefits to growing mushrooms because they can essentially recycle waste products and turn it into something wonderful that we can use for our vitamins and minerals in our diet. And also mushrooms are growing in popularity, and they are expected to exceed the like 50 billion within the next 7 years. So it's a wonderful time to be in the mushroom business. And on the right here is a picture of one of the mushrooms that I work with. And this is, it's Pleurotus austratus, which is pearl oyster mushrooms. So what kind of species am I going to be working with? So far I am working with Pleurotus austratus, which is just pearl oyster mushrooms. And I am also working with Hericium arenaceus, which is lion's mane mushrooms. But along the course of my research, I will eventually be moving towards doing research with, these are Italian oyster mushrooms, and these are pinks, pink oyster mushrooms, and these are blue oyster mushrooms. So why am I using LEDs is because it's low cost. Everybody wants to save money and also save the environment too. So we want to reduce our carbon footprint. So we're going to be using these light emitting diodes to save a bunch of energy, and it lasts a really long time, and they are very safe to use. So just an overall overarching goal of my project is to just look at how substrate composition and LED light colors affect your harvest, how they affect your product that you are growing, as well as but specifically looking at bioefficiency, nutraceutical content, and total antioxidant content. And eventually this hopefully will lead to more development of nutraceuticals and provide nutrient dense foods for people who live in food deserts. So background on how to cultivate mushrooms is just you start with an agar plate. So we do weekly transfers of our species. So we start off with like a little like transfer of, it's just maybe like a centimeter wide, and we let it cultivate and just like colonize throughout the plate. It takes about a week to do that, and then we move on to creating our spawn right here. And so we used cracked corn to colonize our entire spawn bag. And once this effectively colonizes, we move it into our substrate. So our substrate that we use in this project that I'm doing is we did straw and cottonseed, as well as straw and mesquite pods. And once we have this effectively completely colonized, we move into the fruiting production of mushrooms. So which is where you see these wonderful, beautiful caps just emerging from the bag. So just the lifestyle cycle of mushrooms, you start with spores, and spores are right here under this cap. We take the spores and we go through that that wonderful plating, and it goes through like germination. And then once we get to the point where like we're about to start creating these caps, we have it germination, and then it goes to developing this wonderful mycelium. And it's also showing these like little hyphal knots, which will eventually come out of the bag as it is exposed to oxygen and produce these beautiful caps. And it'll go through multiple flushes. So I specifically chose Pleurotus austratus for this project because they are aggressive colonizers. It takes about like half the time as any other species to colonize and produce a cap for people to eat. And I used a combination of straw and cotton seed, and then straw and a mesquite pod. And we put them under red, white, and blue light, and we analyzed the bioefficiency, beta glucan, protein content, and the amino acid content in the first flush, which is the first harvest. And in my experiment, we used 4 bags per for like one experimental unit, because each bag that I use has a really high variance. So what we did was we took an average of 4 bags for a harvest, and just added all that up for one experimental unit. And each experimental unit is separated in 3 blocks and placed under red, white, or blue light. And the way we do it is we harvest the mushrooms right after when it's ready, and then we weigh them, and we use iponic system as well as atomizers for our humidity, and some box fans to have that humidity go around the hoop house that I'm using. So this is just how it's set up. So there's many shelves within each one unit in one block. So this is block 1, block 2, and block 3. This is where you would enter into the hoop house. And so this block is closest to the door, and this one is closest to the evaporative cooling pad. And so they were placed right in the middle of the blocks, and they were also covered with some like black tarps, so that way light doesn't get exposed to other mushrooms that are being grown in the same hoop house. So just very quickly, I did an ANOVA when I first looked at the data of my first 3 experiments. And I discovered that, wow, substrate is really significant when it comes to producing Pleurotus ostratus. And so I said, okay, so I'm going to look at these, but I'm going to separate the LEDs, but because I have conflicting results. So I was looking at each individual species, and I looked at the different colors as well. So when I'm looking at the one with cottonseed, I got around, the highest I got was around like 80% bioefficiency, and which was really wonderful because we want to get as close to 100 as possible. And when you compare that with mesquite pods that we used, they really loved like the composition of cottonseed. So I thought that was pretty wonderful. And so I also discovered that in another one of my experiments, we just looked at the architecture of each cluster and cap size that we got. So in experiment 4, that's what we did. So we looked at number of caps, and we found statistical significance with the red light for a number of caps, and for number of clusters, there were just significantly a lot more red light clusters under the red light than any of the other colors. But the cool thing is, when you're looking at the cap sizes, they were much, much larger when you're looking at the white and the blue light. So these are some of the pictures of the harvest that we did. And this is Pleurotus ostratus that's grown under the white light. This is the blue light. And you can definitely see the difference in the clusters and the cap sizes. There was significantly a lot more caps in red than compared to the other 2. So I also looked at antioxidants, and I saw that in experiment 2 and 3, there were higher amounts of antioxidants in both of the substrates that grew under red light. So I did another experiment just looking at red light, and this is how I set it up. So block 1, 2, and 3, and I did just like the low was just 1 micromoles per meter squared per second, and then the highest is 11 micromoles per meter squared per second. This is how it looked like in my hoop house. This is the top view, and this is what it looks like on the side. So you see those black tarps, they end up closing, and so that way they're not exposed to any other light sources other than that specific one that I'm exposing it to. And so, so far for bioefficiency, I didn't see any statistical significance, but I have not analyzed the samples yet, so that's going to be a part of my next steps. But so far, nothing yet for bioefficiency, but hopefully something with the analysis for antioxidant composition. So, so some of the conclusions that I came to in my project is that Pleurotus austratus really loved the straw and cottonseed composition of each of the substrates that we used. And there were significantly more caps that grew under red light than white and blue, but also white and blue lights influence cap size. And however, there's not enough evidence right now to say that LED light affects bioefficiency. So my next steps are to do more experiments with LED lights and bioefficiency. So next steps, that's what I'm going to do. But I'm also hoping to use blue oysters and pink oysters to see if it possibly has an effect with the color. Maybe it has like a deeper color than if we have them grow under like a lighter and a not as bright light under like red light versus blue. And so that's what I was going to do for my next steps. And I am also specifically interested in Hericium erinaceus, which is lion's mane, because they are growing in popularity, and it's known to improve your immune health and protects your nerves from being damaged, as well as improving depression and anxiety. So promising future for Hericium erinaceus. And I did do one experiment. So the first step in looking at Hericium is to determine, okay, what's the best substrate composition for them to grow. And I did a combination of straw and cotton because that's what worked for another species. But I wanted to see if the carbon and nitrogen composition would affect the bioefficiency. And it definitely did, but it definitely liked oak and soy pellets more than straw and cottonseed, which was very interesting. So far, they really liked oak and soy. And then now I can move towards using light to influence the nutraceutical composition. So I just want to thank everybody for their attention, and hopefully you got some more insight for growing mushrooms. But I wanted to thank my advisor, Dr. Pryor, Bree Rodriguez, the Pryor Lab, and Indigifuse for just being so wonderful and showing it's just wonderful being a part of Indigifuse, and my committee members, Marat Kasira, Dr. Orbach, and Dr. Leslie Gunin-Talaka for being a part of my committee. So I was wondering if anybody had any questions I can answer about mushrooms.
Awesome. Thank you so much, Chrisa. This is a great chance for people to get their mushroom questions answered. And I have a quote from you. I'm going to remember this. It's a wonderful time to be in the mushroom business. So I'm going to quote you on that. That's awesome.
Yeah.
Oh, it looks like there is a question in the chat from Rita. Rita, do you want to read it out loud, or do you want us to read it?
I can just ask it. So I'm not very familiar with mushrooms, but I was just wondering, how would the average person get their hands on some of the different species of mushrooms? When you go to the grocery store, there's only two types, portobello and the white ones, and they don't really have anything else, and so, but I've been reading a lot and seeing a lot about the great benefits of it. And so I was just wondering, how does, general public like me get their hands on some of these different species?
Yeah, that's a wonderful question. So they are slowly popping up at grocery stores that I've noticed over the past maybe year or two. But when I first started looking for these specialty mushrooms, I went to farmers markets, and in Tucson, there's a farmers market right next to the Controlled Environment Agriculture Center, and it like this guy just sells specialty mushrooms, and he has all the species that I've talked about and a lot more. So far that's where I found so farmers markets, and I found some lion's mane at Whole Foods, but it's way expensive. It's way too expensive, and so I will not like get my mushrooms from there, but I tend to go to the like the farmers markets. But like I was at like Albertsons and Safeway, and I finally found some shiitake on the for sale. So that's where I would find some.
Okay, great. Thank you so much. I'm gonna start looking out for them more now that I've had your presentation.
Yeah, absolutely. Thank you.
Awesome. And Cara, do you want to ask your question?
Yeah, hi, Chrisa, great presentation. I'm just wondering if there's a role for, like, does your research help address food insecurity? It seems like you're relatively easily growing these mushrooms. And so I'm just wondering how you see your research contribute to the conversation around food insecurity.
Yeah, so I began doing research and within the mushroom community because I knew that they were a wonderful addition to your diet, but also they can help with your cognitive function, like add some brain juice to your brain, and so that's and they also have a high protein content. So let's just say like there are some communities that don't have access to a good amount of protein as well as other like nutrients that they don't have in their diet and that's not around them. So mushrooms are pretty easy to grow as long as you have a really good system. Like all they really need is a nice like temperature and a good humid environment. And then very, very little light. They don't need that much light. So if we were to establish a system for communities that don't have access to nutritious foods, this can be a really good substitute and addition to anyone's diet to get like your vitamins and minerals in.
Awesome. Any other last questions for Chrisa?
I also want to mention real quick that they also produce a significant amount of CO2. So in greenhouses, I know that plants need CO2. So there's another student that's within my program that's doing they were looking at CO2 production and producing it for algae, for bioreactive fuels, for like space production, for the space, and they just produce a significant amount of CO2. So that's kind of where my research hopefully will lead as well one day. So maybe we don't have to buy CO2 tanks for our greenhouses. Maybe we can just use mushrooms.
Awesome. And there's one last question. Cheryl, do you want to unmute and ask that, or?
Sure. Both Kara's and Rita's question, both of them made me think about kind of the connection to biodiversity. And I know there are seed banks. I was just wondering, is there spore bank? I mean, is there some collection of these different species and, where they can be found?
Yeah, definitely. So I know that at the U of A, there's a professor in the plant pathology program that has like kind of like a spore bank, I guess. And she collects a bunch of cultures and that's what she does on like the 8th floor. And so a lot of people will reach out to her and get a lot of these samples. But also just in the wild too. I mean, mushroom foraging is wonderful. I just I wouldn't eat anything unless like you absolutely know that's the species, because I don't want people to get sick. But you can also find a lot of these species growing in the wild. So like Hericium erinaceus, they are a tree fungus. So you can find them growing on trees like on Mount Lemon. There's a lot of species right now on Mount Lemon. I actually just found out the other day that there's morels on Mount Lemon, which I didn't know that you can actually find, but they're really, really difficult to find. So so that's another way you can get them, I guess, for free. But yeah, you can definitely reach out to people who have a fungal collection. At least that's just where I know so far at the University of Arizona.
Well, thank you so much. This has been so fascinating for both presenters. And we'll be again next week. This series will continue with Najoni. I just wanted to turn it over to Dr. Chief at the end. Carletta, is there anything else you want to mention about this series, your hopes for the series, or the upcoming sessions?
Yes, thank you so much to Chrisa and Luke for their presentations. And I just found it really fascinating about mushrooms. And I just wonder if there was use by indigenous communities a long time ago around mushrooms, but it sounds like a really great option for food security. I know my son has a lot of allergies, and mushrooms like the only thing that is not meat that he can, or not other fruits and vegetables that he can actually eat. So it's just really fascinating to hear about this, especially how the mushrooms grow in the bags. And so thank you so much for sharing this with us. And we will continue these series as a way to connect the students within Native Fuse Alliance, as well as the NRTs at UC Berkeley and University of Arizona. Please join us next week again. We will have 2 more students, one to be determined, but the one who has confirmed is Najona Tallis, who does a lot of work on national parks and conservation of indigenous lands.
Thank you all. Thank you so much, Dr. Chief. This was a great idea. We'll see you all next Thursday at 1 p.m. at the same time.